A very good evening, everybody. Welcome back to our basic module from the Clinical Research Forum. I'm Amartya Sriracha, co-leader of Clinical Research Forum. And here we have Dr. Pavan Kumar CM, a second year of PhD student from Sri Adi Chanchuri College of Pharmacy. So, today in our session, we are going to learn in detail about converting an idea, that is like a research idea, that a hypothesis into an executable project by developing a protocol, which is the essential step of doing a research. Before we move on into our session, I want to take a few minutes to introduce our speaker to you all. Our speaker here is a researcher and he has multiple projects ranging from original research studies and clinical trials. He is a Google Scholar indexed potential researcher. So it is an honor to us to have him here as an expert in the field, and it's a pleasure to hear from him and learn from him. So without any delay, let's hand over the session and learn from him.
Yeah, thank you, Amatya, for a brief introduction. So, as you all know that I'm Mr. Pawan Kumar CM. So today I'm just going to speak about the topic that is protocol development and core components of the research protocol. So before going into the concept, I just want to talk few words regarding clinical research. So when we talk about clinical research, the first and most important concept is planning. So clinical research is not a trial and error process. It involves human participants, research, and also ethical responsibilities. And a research protocol serves as a blueprint of the study, just like we can't construct any kind of building without a blueprint. And the protocol ensures that the study is scientifically validated and meaning the objectives, methods, and outcomes are logistically and logically conducted and connected, and also it in the same time ethically sounds, and also it ensure the participants safely and rights are being awarded for them. And for the regulatory perspective, a protocol is mandatory. No ethical committees or regulatory authorities will be approving a study without any clearance of this clear protocol. So why do we need a clinical or a research protocol? First and foremost, the clinical research must be planned. And as we talked, it should be ethical. And also it should be responsible and also reproducible. And a clinical research protocol acts as a blueprint for the entire study and also ensures safe, scientifically validated, and the participants' safety is very important. And coming to the ethical committee, we are going to get an ethical committee clearance only if our protocol is maintaining a standards, just providing patient a safety and a standard ethicals, and also mandatory for ethical approvals and regulatory compliances. And maintaining the consistency and transparency is very important, and maintaining the quality of a research till the end is also very important. First we are going to generate an idea, then we are going to get a protocol. Like in a team we are going to prepare a protocol, then we are going for the ethical approval, then the study will be conducted, then we are going to the results. And coming to the research uh, protocol, it's as we all know that it is a blueprint. And then the research protocol is a detailed writing. Like a detailed writing means five core pillars are there. So that five core pillars are like written master plan, a detailed written document detailing the study end-to-end strategies, like how we are going to start and how we are going to stop, and what are the hypotheses what we are having. And in the methodological framework we can just see what we will be doing and why we will be doing and how we are going to do. And coming to the operational definition, clearly defines terms, roles, like a team roles, and also the responsibilities of the team, and also the clinical procedures, and also measured outcomes. And as we all know that ethical structure is very important. A scientifically structured document ensured participants safety and data integrity. And not only conducting the research in a safer and ethical way is important, and also we have to comply to the regulatory authorities fully aligned with the ICH GCP, which is good clinical practice, which is designed by WHO and global regulatory guidelines. So coming to the research idea, we just used to hear what is research idea, what is a research proposal, and we were having a different kind of words, and we are just having a confusion related to what is a research idea or a research proposal and research protocol. So in simple terms, research idea is just an initial concept or a question, and research proposal is a plan prepared to seek approval or funding, and research protocol is like finally detailed operational documents and protocol including complete methodology and ethical details, and once approved the protocol must be strictly followed. And in important way, I just want to conclude this slide with one word, that is like, once a protocol is reviewed and approved by the ethical committee, it becomes a blinded document, and any deviation from it requires justification or approval. We can't change any of the things which we have mentioned in the protocol just after the ethical clearance, and therefore the protocol is not only just a plan, it is a commitment. As we have shown in a triangle, it have a specific values, and research idea is a simple that starting point. It is through the observation or clinical question that arrives from the patients or literatures, and research proposal is a development to consistency. It is usually submitted to the institution or a funding agency or an academic committees. And coming to the from research question to protocol, how we are going to design. So here we can just say like, identification of a clinical research problem or a clinical problem is very important, and then we should go in depth like review of existing literature, whether we have any of the existing literature related to the topic, because simply if we are doing a research, it nowhere having the value, it's simply waste of our time and a resource, and also economically it's a burden. So if we are planning any kind of research, it should be having some kind of novelty or a uniqueness or something related new, and which is also beneficial to the society and which is also useful for the society. And identification of the research gap, as we discussed, already we should see a literature search already, whether we have a same research what we are going to conduct in the previous study, and also we have many websites like Shodhganga and all. We can just put a title before we are going for the ethical clearance, that whether the same title is being exist or same research have been already carried out, and after confirming there only we can just go for the ethical clearance. As we told that once after the ethical clearance is done, we can't change any of the title or a topic, but in kind of PhD research or somewhere like that, we can change a title or an objective with certain rules and regulations by paying a penalties. And framing a clear research question, a clear research question is very important. So once if you are keeping a title, it's not done, and once if you are keeping an objective, it's not done. Framing a clear research question is very important, and translation into a structural protocol. Like a structural protocol means, in further slides we are just going to discuss in depth. Like a structural protocol means it should have a very clear introduction, and where the introduction should be in a format like, in a starting we have to mention a clear structure, what is the research, and then we have to speak about the percentage or an epidemiological way, like in a percentile way, coming to a global or a national or an international. Like on, we have to say in a percentage the prevalence, then we are going to interlink the title, and we have to write a structured introduction. But in the end of the introduction para, where we are going to mention what is our study, exactly what we are going to do, and it should be in an introductory way, not in a methodological way or methodological procedures. And we are just going to use FINER, FINER criteria for research question. F mean feasible, and also next is I means interesting, and N means novel, and E means ethical, and R is relevant. So one thing we have to focus if we are doing any kind of study, or if we are doing any kind of research, like whether we have to check for the first feasibility. If I'm taking any kind of genomics research and all, and if I have to do in any of the local hospitals or local laboratories, it is not good enough laboratories what I have. So that what we are planning, where we are planning, and what we have to do, whether we are feasible to that, we should just consider first. So that we have mentioned that FINER criteria, that is feasibility, and also whether it is having any interesting research question. And also we discussed that novelty, whether we have something unique, whether we have something which others have not done, whether we have doing any kind of research gap which already is not existing, that comes under novel. And ethical, like being a pharmacy practice or related to healthcare science, it is very important to adhere for the ethical committee or ethics, because we are daily dealing with the patient's lives. So according to me, ethical will be the first priority, and then relevant, whether the topic or a title, whether the protocol is designed relevant to the title, or whether it is structured in a relevant to the objectives, will be very important. Then coming to the types of research protocols, like many kinds of protocols are there, different types, but we here I just mentioned only five which are basic. Like one is observational study protocol, and one is interventional study protocol. Then comes clinical trial protocol from the phase 0 to phase 4. Here I have mentioned phase 1 to 4, and you all know that the phases, I guess phase 0 is microdosing, and phase 1 is human pharmacology, and phase 2 is therapeutic exploratory, and phase 3 is therapeutic confirmatory, and phase 4 is post marketing surveillance. And also academic, like investigator initiate studies will be seen, and also industrial sponsored studies will be there, like coming under clinical research. Or even though if you are a PhD student or any kind of basic researcher, you can just collab with any of the industry person and you can just carry out the your study. And study designs are as we have discussed, and in the observational study protocol involves only the observational outcome actively intervening, and this includes cross sectional, cohort, case control studies. They are commonly used to study diseases pattern, risk factors, and outcome. As the name itself, it is indicating that it is just an observational. By observational, what you are just going to see. And coming to the interventional, like it involves actively introducing an intervention such as a drug, might be procedure or a educational program, and assessing its effect. This requires strictly ethical and regulatory insights. When you are just going to change, or when you are just going to suggest any doctors or any kind of person to change or to modify anything, it will come under the interventional. And as we discuss the complete phases, it will come under clinical trials and academical study, what we are going to conduct in a health sectors, like in PharmD or a farm pharmacy practice. We are going for the ethical clearance. And also this comes under industrial, like academical studies. And as we discussed, we can collaborate with other industry persons if we are more interested in a research. It will be in industrial sponsored studies. So core components of research protocol. Let's discuss. So core components are like administrative components or scientific components, and the methodology, scientific components like ethical and regulatory elements, operational and analysis plan. If you are going in depth by one, like administrative components, like define who is responsible for the study and where it will be conducted, it comes under administrative. And the scientific components is like justify or justification why the study is needed and what it aims to achieve. Like simply if you are doing a research, it's not so good. As we told, it is just a waste of time. So it should have a clear aim to, and what we are going to achieve should be mentioned in the protocol. And coming to the methodology, explaining how the study will be conducted, this is a backbone of the protocol. So protocol is a backbone of the clinical research, and a methodology is a backbone of the protocol. And ethical and regulatory elements like ensure patients participants protection and the legal compliances. And now we are going to just structure, let's see about the structuring the research protocol. And in the title page of the protocol, we are just going to mention title of the study, name of the investigators, like whether we are under anyone, like we are going to give our name, and also the guide name, or any other people or any other industrial person or other laboratory persons who have just collabed with us, who helped us, and who are equally contributed their works like us, and we are going to share them authors. And here if we are going to a research protocol, here only we can mention our name if we are doing under our research, name of the investigator, and also the institutional affiliation should we have to mention. And also protocol version is very important, because as we told, like a protocol, we can't change any kind of things once we are went with an ethical clearance. And once if we are need to change anything, again we have to give a justification for the ethical clearance or ethical members, so that there we are going to change the protocol version and the date. And also sponsors, if any of them we are having, we are going to mention. But in an academical things, we are not mentioning any of the sponsors, only our name will be there, and also under whom we have did, that will be there. And the image is not so visible, I guess, so where you can just see the page numbers and the page title, how it will be, and where the author name should be coming, and also the course, like which course we are just doing, and also the due dates and the instructor date, and also the affiliation should be mentioned, and the investigators and the study sites. So it is important that once if you are planning with the protocol, after writing an introduction, then we are going to write a need of the study. In the need of the study, as we all told, we are going to consider this FINER criteria, and we are going to write a need of the study. Then we are going to write a literature review, like literature search, like in which author did what research, and where he did, why he did, what novelty he did, and what research gap we have identified, and from that what we got to know the research gap. Those everything will come under this. And here study sites we are going to speak. And in the study sites, we are clearly mentioning that principal investigator is nothing but a doctor, and co-investigators, this protocol is bit related to a clinical trial kind of, because in our normal academical way we are not going to mention the principal investigator, because as a name itself is ours. So we will be the principal investigators and the co-investigators, and the study sites where we are going to carry out the research, and the roles and responsibilities. What are the roles and responsibilities we are going to carry out, and what each person is going to carry out. And also the qualification and the experience matters a lot. If our guide is having the capability to guide us and to also to manage the things, so it will be well and good, or else it will be struggle for us. And coming to the background and the rationality, background and rationality section explains why this study should be conducted. And as we discussed, like a need of the study, this is one of the most closely evaluated sector by ethical committees, reviewers, and the regulators. We begin with a brief overview of the disease, or a conduct like a brief disease or any conditions, disease conditions, only what is necessary to the set of context. This is not a textbook description. Like here we are going to clearly speak about what is the background, what we structurally writing in the introduction. It will be like overview of the disease or conducts and current standards of care what we are having, and as we discussed again the summary of the relevant literature, then the identification of the knowledge gap and also the research gap, and we are just going to give a clear structure of a scientific justification for the study. And here we have, I am just showing that scientific justification, a research funnel. And first we can see that disease overview and standard of care, like how the disease have been prevalented and how the globally it is having. If the disease have already been solved and it is not in a society, it's not a good thing to carry out that research, and it should have also a standard of care should be there. Then the literature synthesis should be there properly. We have to check for the existing literature and how they have gone through, and we have to identify a research gap, identifying the knowledge gap, so that what additionally and what novelty I can bring out from this research, and also scientific justification under study hypothesis. In further slides we are going to see about the study hypothesis, and study hypothesis is also very important while planning for the protocol, because what you are going to expect in the end of the research, it is nothing but a hypothesis. So objectives of the study are like primary objective and a secondary objective, and the primary objective will be completely related for your main title, and also secondary objective will be like exploring additional outcome related to the primary objective. This provides supports or complementary information but are not the main force, main focus, and the main focus will be primary objective only, and exploratory objective are optional and are usually included to generate hypothesis for future research rather than the draw identification conclusion. So it is nothing but like once we are just carrying out any kind of research, some of the standard ways are there, leaving a thread of the research. So someone who reads our research, someone who is going under our research, or someone who is so interested in that field, so if they are going through our research, so he will be identifying one of the research gap, and or else we could only give a thread for him, so he's just going to carry out that thread and he's going to conduct the new research which we have not done. So objective must be very clear and measurable, and objective guide methodology and analysis is also very important. And coming to the hypothesis, we are so much like familiar with the null hypothesis and the alternate hypothesis. And null hypothesis is that no effect statement, and alternative hypothesis is pre-detection of a statement. And null hypothesis also like assume no relationship exists between the variables, and also it is denoted with the h naught, states that there is no difference or no association before variables. This is the hypothesis that is actually tested statistically, and also it aims to the reject or fail to reject this. And coming to the alternate hypothesis, it is an assumes a significant relationship or differences exist. The outcome the researcher expect to find and aim to find evidence to the support this. So in a clear image, what we have mentioned is like a solid question should we have to frame, and what makes a good hypothesis we have to plan. Then we have to go for the background research. Then we have to make sure it's testable, and as we discussed, whether it's a feasible. And next one is independent and dependent variables also we should find, because it is a very important for the hypothesis. And study design, if we are planning, there are many study design what we can plan, like a type of study designs, study setting, study duration, participants flow, overall study flow. Like the study design describes the overall structure of how the research will be conducted. It is one of the most critical section. And also the protocol, because it is determine the variable and reliable of the result. And also the coming to the types of study designs, for example, cross sectional we are going, and cohort we are going, case control we are going, randomized control trial are there. So randomized control, randomized placebo control is a gold standard in a clinical research. And also because it determine the validity and also reliability of the result. First, we specify the type of study. For example, as we discussed, the many kinds of studies, that is cross-sectional, cohort, case control, randomization, and also the quasi experimental studies also we are having. And the study settings will be like study setting describes where the research will be conducted, and such as hospital or a community or a outpatient clinic or a laboratory. This helps in understanding the context and generalizing the finding. And study duration. Study duration includes both the total length of the study and key phases such as recruitment and investigation, follow up, and analysis. So here I have showed in a simple way that once if we are designed with the protocol, here we are going to write how we are going to screen, and that is inclusion and exclusion criteria, how we are going to screen, and what are the responsibilities and roles are there, and how we are going to enroll, and intervention, what we the intervention we are going to do, and whether we are what any changes are there. That is nothing but a follow up, not just taking a data of a subject today and not following up. So we have to specifically follow up for the each patient, and analysis is very important at the end of the day, of the end of the research. So these all things should be mentioned clearly. And coming to the study procedure, here is a study population. So where we are going to take one of the study population, and in that we are just going to, this is a sample what I just planned. So in brief out, I'll just explain. Once we are taking a study population, we are just going to make a two groups. One is controlled group and one is a IMP based group and all, so that we are going to see what the effect is coming out of the control group and with the, in the IMP group. So let's think like in a group we are going to give IMP, and in a B group we are going to give any kind of placebo controlled. So here in placebo control we are not going to give any of the um, investigational medicinal product things or any standard API drugs and all. It is just a placebo, a dummy. So that here we are going to carry out or going to see the comparison between the who is taking a standard care and who is taking a placebo or an IMP based. At the end of the day, we are going to see the result. And participant selection way is very important. That the inclusion criteria and the exclusion criteria, maximum these things we are going to mention in all the criteria. We are going to see in all the research things and all. That is age in between 18 to 16, and a diagnosis with so and so disease, and who is going to provide informed consent. And coming to much more related to that, if you're going to do any of the cardiac things in specific to angina, so we are going to add only angina subjects, like that we are going to mention. And coming to the exclusion criteria, like a majority we are going to leave the pregnancy and the lactation women's, if we are not having a clear picture of the research, and serves a comorbid conditions if they are so severe, and concurrent treatments are going on to the person. So other key consideration are like vulnerable population, like example, children's, and recruitment strategies, and esteemed sample size. So there we just talked about esteemed sample size. So once if you are going for the sample size, we have to see that determine total participants. First we have to see whether like how many population surrounding we are having, calculate the exact number of subject required for the population, and also ensure study power, provide the statistic strength need to detect a true effect in a sample. And in the clear picture we have seen that the whole population is there in that sampling frames what we have seen, and in that what we are going to take a sample. And many kind of sample sizes are there based upon the study things. And a secure result reliability, minimize the risk of false negative or false positive. And also the factor drives calculation accounts for expected effect size and data variability. Regulatory requirements also we are going to see. Regulatory requirements a mandatory justification for ethical and scientific approval. And also as we discussed, once we are getting a ethical clearance also, we can't change any of a plan. As we discussed, it is a not just a plan, it is a commitment. And finally the sample size must be the feasible and ethical. Investigator must balance scientifically rigor with the participants safely and resource availability. And here comes the intervention or exposure type of, well, here we are going to see the type of interventions. And as we discussed, type of interventions are like based upon the drugs or the procedures, whether we want to alter any of the things, or whether in our observation kind of. And in a dose adjustment, this ensures that the intervention is consistently across participants and reproducibly in further studies. And for coming to the duration and the frequency, it is just an applicable for the drugs and procedures, a detail to that dose duration and frequency. This ensures that the intervention is consistent across participants and reproducible in the future studies in related to the same meaning what we are going to see. And also the comparator or controlled group, as we just seen in the study design, also in the picture what we discussed in the previous slides. Also we are going to give two kind of study sample, and one is control group and another one is like experimental, like who is going to take a API drug or a standard drugs or a investigational medicinal product drugs. And standardization and monitoring of intervention is very important, because once if you are giving the placebo or an IMP drug, it's not like okay we given, and if you are not following up also it's fine, because it's our responsibility to stand on our ethics, that is safety and efficacy. And a safely consideration is very important. We should look after the patient safety first. And the in the protocol it is also very important to write the outcome measures, which also in another word we can tell like mm, outcome of the study, what we have mentioned in the need of the study, the same result what we are going to see in the outcome of the study. And also we can just say that it can be in a hypothesis. And sorry, endpoint type, how the endpoint types, like a primary objective what we have kept, and the main question the study is powered to answer, like reflects the main goal of the study and are the basis of sample size calculation or an hypothesis testing. And coming to the secondary outcomes, it provides the additional information and may explain the results or explore related effects. And coming to the safety outcomes are essential to monitor any adverse effect particularly in interventional study. They protect participants and fulfill regulatory compliances. And here, the clearly defined tool and scales are measurement. Like example, cognitive functioning may be measured using the MOCA. And here I have just mentioned tool and scale standardizing metrics. Like if we are going to see any of the diabetic things also, where standard tools are like we are going to check for HbA1c or MOCA, that is Montreal Cognitive Assessment. These are like very basic and standard things we have to just look after, because the tools stands a backbone for the methodology. So data collection methods. So here only we have mentioned about the case report form, but in standard we can mention many of other data collection forms, like maybe through the case report forms, source documentation, standard operating procedures, and also like a data monitoring and verification, and confidentiality and data security. These are the like key principle should be there in a researcher mind before he is going to conduct any kind of research, or when he just going to plan for the a research protocol. And data collection form methods should be like in many ways are there: clinical case report, case record forms, what we have mentioned here, or we can go through the intervention, like having a interaction with the subject, and also by the data collection standard tools, all the standard questionnaires or standard preparation of our own questionnaire. And then we can go for the copyright if we are want to make it more standard. And it's not so mandatory that to go for the copyright, or if you are going for the copyright, it should not be like motivational for others to go for the copyright, because it's not a good sign of a researcher. And this is more important that like a pharmacy practice or any kind of healthcare sectors will be much far from the statistical analytical or analysis plan, because this finds very difficult for the researchers in related to healthcare. So it is like descriptive analysis or any kind. So in descriptive analysis, what we are going to see is a mean, that is numerical average, and a median, which is a midpoint. I just don't think that we have to go in depth related to what is mean, median and all, because where we have already studied those in our previous classes itself. And also the percentage ratio or the fraction of 100, and a percentage frequency, like number of occurrence, and a range, highest and the lowest. And coming to the inferential analysis, where we are going to see the correlation regression analysis. And regression shows as predicts the relationship between two variable. And analysis of variable variance are like testing the extent to which two plus group differs. And ethical consideration, that ethical consideration are mainly focusing on the informed consent. Before we are going for the informed consent, we have to know about how we are going to take a informed consent. And there are so many in-depth criterias. If you are going to take a informed consent, it should be in a four walls, which should have a poster magazines related to the clinical sectors. And while explaining also in a normal academic procedures, academical research, where the PI is going to explain. But when it comes for the clinical trials, the PI will be anyhow the doctor, and doctor is ensuring the informed consent whether the patient or a subject have understood the research completely, and risk benefits assessment. Before going for the research or before enrolling, we should make him aware what are the risks and what are the benefits which we are going to provide, or which he is going to get a benefit. And confidentiality and privacy. Before doing any kind of clinical trial, the director of the clinical researcher is going to sign for the confidentiality disclosure agreement, because to maintain the consistency and a confidentiality of that subject and participant rights. And wherever you are seeing a clinical research, it is clearly mentioned that a patient or a subject can leave the study at any time. And vulnerable population, coming to the vulnerable population, applies exact extra safeguard for protecting or sensitive groups, because in the clinical trials what we are going to see, till the phase 3 only related to disease specific. We are not going to take a comorbid condition patients or something who is so severe in the nature. So once after it is marketed, we are going to see in a vulnerable people. So that vulnerable population also we have to focus in the protocol. And the key principle is transparency should be there, open communication throughout the study lifestyle should be there, accountability that we should adhere to the institutional or in independent review board standards. We have two, that is in the institutional review board or an independent review board. And end of the study and dissemination, the 5-step strategy flow works are like a study competent procedure we have to mention, and data cleaning and final analysis should be we have to mention, and reporting and results how we are going to do is very important, and publication and presentation is very important, and authorship and dissemination ethics are also important. Before going to all those things, we have to make a specific research in a Wikipedia blog, social networks. Nowadays social network is just booming like anything, and university websites we have to see, and preprint archives also we have to check. And quality assurance and safety monitoring, how we are going to structure a protocol is not so easy. Like quality assurance also it should match, monitor and control should be there, identify issue with the process should be there, and generate corrective action should be there, verify and implement corrective action should be also very focusedly keenly have to be observed. And protocol deviation and correlation, what we can see in this slide is having a main role. If we are doing a follow up, and if the next visit of the person is in a this week or in the next month, and if he is not coming to on time, or if he is not taking a drugs in a proper way, or if he is not adhere for the medication, then we are just going to make it document that is protocol deviation. And if we are mentioning any kind of unnecessary things in a protocol, there also we can just make it as like a protocol is deviated from the title or something out of the boundary what the title and the objective stands for. And protocol deviation should be very clear that whenever we are going to make a report or a document of a protocol deviation, and in a clinical trials it's a gold word, that protocol deviation. And study documentation is very important. Research protocol, informed consent form, and case report form, and also investigational brochure manual, and data management and records, how we are going to plan all these things, and how the study or how the research documentation we are just going to maintain. These are the standard ways, and how we are going to data securities and managements we are going to provide, and the confidentiality we are going to make, and privacy we are going to keep. These are the key things, key principal things which we have to just make sure and speak in the ethical clearance. And these are like common mistakes in the protocol writing what we are going to see, maybe spelling, punctuations, and grammar things what we are not focusing. Just we are focusing on the research, but when a standard researcher is going to validate your research, we have to consider all these things. And also the style and form, and also the how we are going to include continuous error or continuity errors we are going to stop, and problems to the cohesive cohesion and pacing we have to look, and also the tone and voice is very important. It should not be in a very harsh or very hard way, and we should write in a very polite way how we are going to carry out. And the reputation is also very important, because the affiliation stands main and more, because if you are doing any researcher under any of the industry person collaborating or any kind of clinical trial, it is very important to see the reputation of a researcher and also the corresponding author, and also to the institution or to the industry. And tense, like a clarity should be there, and the narration should be proper, and the logic and arguments should be very clear, need to show a clear sense of connects and cause and effect. Because once if you are keeping a title something, and if you are making any kind of different objective, it nowhere related. So that logic and argument should be in a proper way. And these are like subtext contents, and formatting should be in a proper way. These are the basic things what I have mentioned. These are the very basic thing, 10 common writing errors what we are going to speak, or what we are going to do in structuring the protocol. And the key take home message of this like a protocol session is like, protocol is a backbone of the research, and a clear objective driven methodology should be there, and ethics and science must go through in our research, and accurate data and analysis is critical. And currently we are having many kind of statistical tools like SPSS version 30.0 is currently what we are available of. And if not, if you are a Mac user, or if you are a even the window users can go for the JMP, which is having a very vast things and all. You are just going to can make any kind of graph using this JMP, which is very booming now. And also the documentation ensure transparency should be there, confidentiality should be there for the subjects, and the privacy should be maintained, but documentation transparency should be there for the society. And here I'm just going to conclude my slides. That is like again the take home message is like, protocol as a foundation. A robust research protocol is a central pillar of a success, ethics and responsible and reproducible thing what we are going to see in our research. And it should be a scientific rigor and ethical standards we should match, and data in integrity should be there, and also the transparency what we have told should be there in our research protocol. So protocol is central to success, ethical research, clear methodology and objective ensures reliability. So here just made all the things clear what I feel. So if you guys are having any of the doubts, please put it in a chat box so that we can have a conversation there. And hopefully I can just maximizely I'll try to clear your question and discussion.
Yeah, we are here at the end. Thank you so much, Pawan Kumar CM, for such a wonderful session. You began with the basic terminology, clearly defining and explaining the differences between research idea, research protocol, and research proposal. You took us through the each and every step of process of developing a research protocol, and you went in detail with different parts that are required to make an ideal protocol, how the goal should be, that is to not waste the resources and time, and the ultimate purpose of research is to fill the knowledge gap. So yeah, I'm seeing here in the chat box, we have a couple of questions.
Yes, please.
One of them is a very genuine question, which is how much literature review is enough?
Okay.
Are we underdoing it or overdoing it? Do we have a clearly defined criteria? Do we have to follow any timelines? Like, should I go back 5 years, 10 years? How much literature should I review before I arrive at my research question, that is, research hypothesis?
Yeah, very good question. Because nowadays, even with my friends or with my juniors, I used to discuss about this because they used to bring me like a 2021 or 2010 program things and all. But according to me, or according to my friends or my seniors or my godfathers, what they're going to do, I'm just going to follow up those things. I used to suggest only go through just back of 5 years, because everyone should have a novelty, something unique, so, and which is latest to the society. Because one, if you are going so back, like already leprosy have been just washed away. But now if I'm going again for the same things and all, it is not so good enough. So we have to just go with the recent articles, just before this 5 years, very latest version. And here I'm just going to suggest one thing. So as we have talked about the methodology, like MOCA, Montreal Cognitive Assessment Test, or might be, or something like a nicotine dependency, what we are going to use, like a Fagerstrom nicotine dependency questionnaire and all. So once if we are going to take any kind of this kind of questionnaires and all, we are we have to take a original review itself, original research itself. So once if we are going to take original research for that questionnaire, might be it is in a 2000 or it is in the 90s, like we have to take it, because we can't change like any of the Newton's law. We can't say like Newton's third law, I will take in a latest version, like past 5 years, because Newton's third law we can't change, or any kind of standard protocol or questionnaires we can't change anywhere. So when we are going for those things, we have to go for the original, let it be any kind of year. But when you are just going for the research gaps, research knowledge things, I suggest only to go with the last 5 years. And also one more question, as you asked, like how many articles or how many literature search is very important. It basically depends on what you are doing, because related to pharmacometeorovigilance, we'll be having very less in a number. And if you are going to any related to diabetic, it is almost reached the cell level, so you are going to get a very vast number of articles. So it depends, but according to me, I used to suggest, if you are going to design a protocol, go with the at least of 30 to 35. Because once if you are going to structure a manuscript for your thesis work, you are going to keep around 45 to 55 according to the standard way. So if you are going to the 30 to 35 in a protocol itself, it is very useful for you only to do a research and to know the very research gap before you are going into the research. So I used to suggest go with the at least of 25 to 30 before you design a protocol.
Yeah, yeah, that actually answers the question. So we have another one.
Yeah.
In your experience, what aspects of informed consent is needed to be mentioned more clearly, in the
Mhm.
sense that what aspect can be frequently misunderstood by the participants? What
Okay.
should be focused more when you are writing a protocol?
Okay, here I could see two kinds of question. One is informed consenting. If I just answer in a single way for the both the answer, yeah, like preparing a CRF or a case report form or a informed consent is very important way, and we have to suggest like what are the risk and what are the benefits what they are going to get from our research. And also if it is a clinical trial based, where we are going to give, where the sponsor is going to provide us some kind of financial support for the visits. So here, before giving a informed consent, there are 10 standard criterias which should have mentioned before the enrollment. And this is done by the PI, that is a doctor, where they are going to say that the patient can or a subject can enroll for the study and he can leave at any time he want. This is the thing where the PI have to clearly mention, because if you are enrolling for that any kind of clinical trial, research things and all, the already the subjects will be having a fear whether we are taking a placebo or whether, because it is as if many kind of blinded studies are there, if you are so aware. So patient, if he's knowing, it's fine, or else if he's knowing also, without knowing also, if he's taking any of the IMPs or a placebo, he will be having somewhere the feel of whether what can be happen tomorrow for him. So that it should be very clearly briefed out by the PI that what benefits he's going to get, and he will be getting complete hospital charges from the sponsor. And if in case any of the serious illness, and if he is going to expire, it is very clear that company is going to pay some kind of amount for his family, because before signing for the informed consent, we are just going to have a conversation between the subjects and any of the responsible person with the subjects. So these are the things which we have to clearly mention. But max to max, we will be seeing the benefits what in a clinical research or in a kind of trial studies, because where the subjects will be getting a thing in a free, and also the sponsor is going to pay them for the visits. So these things, I guess, we have to stress out, and we have to make the volunteer more aware, and we have to reduce the person ideas like what might be happening tomorrow, because the whole charges will be taken care by the sponsor, because nowadays we are going to see the financial burden more. So these are the things where we have to stress out.
So yeah, it's pretty much that we make them aware that we are here to help you out if anything happens. We can provide you the backup, you need not be scared, and also make them aware of what are the consequences that are going to happen. So
Exactly.
yeah, thank you for the answer. We have quickly another question, if you
Sure.
don't mind.
Yeah, please.
Yeah. So you have mentioned common mistakes to avoid, like the 10 common mistakes to avoid while we go on writing a protocol. So we have a question here that says, do you have any an advice for the beginners on a foolproof approach to writing a protocol to avoid rejections and amendments? Of course, you have mentioned all the steps of writing a protocol, but just like maybe a quick brief for a beginner.
Okay, so basically, if you are a pharmacist or if you are a researcher, we will be not so much concentrating on the grammar things and all. But if a standard researcher is going to read our research, it should be very in a way to reach out. And if there are many software tools like AI tools or like a Grammarly and many other AI tools are there, which is going to generate or make our research in a next level. But if we are not going through our protocol, it is very difficult what we can justify. So before doing any kind of, or before going into any of the AI tools, just read it out, understand yourself, and after the AI things or the AI correction, please go through the AI, like a corrected AI part, because man itself created the AI, so anything can be done there. So before you just give a protocol to your guide, first you understand what mistakes you have done.
Yeah, that is a very good answer. So, yeah, if you guys have any other further questions, you can put up in the chat box and reach out to FCPI and also to Pavan. That's it for today's session, and hope we will meet you for the next basic module. Bye. Thank you so much, Pavan.
Yeah, thank you, Matya.
Bye.
Bye.